And thank you, Charla, for reminding me. I always lean on my students to remind me to record. I always forget. Okay, we're going to get started. It is my absolute pleasure to introduce to you today Ryan Besemer. Ryan grew up in New York. He has already had an impressive science career for someone his age. He started conducting research early in high school, and he is already a published author in the journal Viruses. He has researched viruses in isopods, sea star wasting disease, and now hypoxia and corals, and he'll tell you about that today. During his time at UNCW, Ryan has only majored in marine biology, environmental science, minored in chemistry, minored in statistics. Clearly, he's a real slacker. I've had the pleasure of also having Ryan in my class. He was part of my coral reef ecology class in the field course where we went down to Belize. And it was just so much fun having Ryan on that trip. During that trip, that's the first time Ryan surprised me, and he always continues to surprise me. So we attended this local drum demonstration where the drummers were encouraging everyone to get up and dance, and no one was moving until Ryan popped up and pulled a couple girls on the dance floor, and that just had everything started. Everyone else followed, and it was one of the highlights of the trip. Just thinking about it just really still makes me smile. The other time Ryan surprised me is learning for his love for hockey and playing hockey. And then I recently found out another fun fact about Ryan, and I can't spill the beans just yet. I'll let him do that. But Ryan has just been an integral part of the laboratory since he joined about 1.5 years ago. He's volunteered, he's conducted a DIS, and then also conducted his honors thesis. He's logged over 250 hours in my laboratory, creating the seawater system, doing everything from painting and getting that up and running. He has just been unbelievable. He's helped with the creation of the seawater system, maintaining it. He's helped others with experiments and then conducted his own research as well. Ryan has juggled an impressive course load through his career at UNCW. He's also conducted research. He has a lot of extracurricular activities, and he still works through all of this. So it's amazing. He's a very bright young man with endless potential. He has been a joy to have in my laboratory, and I look forward to following his scientific career because I have no doubt will be remarkable. Before we get started, I just want to give a quick acknowledgement and thanks to his committee, Drs. Chen and Dr. Tift. And without further ado, Ryan, please go ahead and tell us about your research.
Awesome. Well, thank you, Dr. Fogarty, for the very generous introduction. I welcome everybody to my honors thesis. And today I'm going to be talking to you about protein reserves in a facultative symbiont coral species, Oculina arbuscula, after hypoxia exposure. So as many people know, we are seeing changes within our climate, and that can include changes in rainfall patterns, increases in temperature, and even changes or the occurrence of stratification or layering within the world's oceans. And another understudied threat that is also being contributed as a result of climate change is what we know as ocean deoxygenation. And so this figure here shows the changes in dissolved oxygen concentrations throughout the globe. And while you will see some areas here in blue indicate where we are seeing increases in dissolved oxygen content, you are also seeing a vast majority of areas with red where we're seeing massive declines in dissolved oxygen content within our oceans. And so for the most part, we are seeing major declines in the dissolved oxygen content of our oceans. And with that, there are several major drivers. The first driver to ocean deoxygenation is eutrophication. And eutrophication occurs through the excess input of nutrients as a result of runoff and agricultural pollution. And through that, we see an increase in phytoplankton blooms and all sorts of an overall increase in respiration as a result of this addition of nutrients and added organic nutrients, which then depletes oxygen levels through respiration. Another role in driving ocean deoxygenation is temperature. And temperature has several different ways in which it can impact ocean deoxygenation. but the largest contributor is likely through its decreased solubility of gases as temperature increases. So oxygen, being one of those gases, will decrease its solubility in substances of higher temperature. And so as a result, the water or oxygen holding capacity of waters, especially sea surface temperatures, as we've seen a large spike in sea surface temperatures, declines as we see climate change continue. And then finally, stratification, which stratification can also be linked to temperature, but there are also several other roles that result in the stratification of our oceans, and that can include a result of differences in salinity within water masses, as well as density differences in water masses. And stratification is likely to be one of the largest contributors moving forward to ocean deoxygenation in the coming decades. And stratification, how that results in ocean deoxygenation is through this layering of water masses and the deplenishment of oxygen in deep water masses. We see that these deep water masses are unable to replenish their oxygen due to a decreased vertical mixing as a result of this stratification. And so, as I mentioned, all of these are contributing to declines in dissolved oxygen content within our oceans. And now ocean deoxygenation is also what we call marine hypoxia. And this is defined as less than or equal to 2 to 3 milligrams of oxygen per liter. And so I really like this figure because it does a great job in kind of depicting the importance of this current study we're looking into. So all of these red dots you see here on the figure are what are known as current documented dead zones. And a dead zone is an area scarce or devoid of marine life as a result of hypoxic conditions. And now, in addition to that, you are also seeing all of these blue and purple areas where we currently see coral reefs present. And with that, you see quite a bit of overlap as well between the documented dead zones and the area where coral reefs occur. And then finally, the really interesting point is hypoxia has actually already been noted to have an implication in coral reef mortality. And these yellow dots show the areas where that has been noted. And so seeing that marine hypoxia or ocean deoxygenation is such an understudied threat, and even further understudied on coral reefs, the understanding and importance of understanding hypoxia on coral reefs is that much more important, especially because coral reefs are such a highly endangered habitat. And so now to look into some of the previous research done on hypoxia in coral reefs. There are very few studies, actually only 8 in total, but I wanted to highlight 2 here. Of those studies that have been done, even fewer of them are actual experimental studies. And so that's why I wanted to highlight this first study, because of the experimental studies, it is the longest one occurring of those studies. And going along with that as well, most of the experimental studies done on hypoxia in coral reefs are on tropical coral species, at least in terms of the experimental studies. So this first study that I've mentioned here from Haas et al. 2014 is of Acropora youngii, a tropical reef building coral. And the oxygen tolerance that they found in this paper was at 4 milligrams of oxygen per liter. And the duration of this experiment was for a total of 10 days. And so in the results, like I mentioned, the oxygen tolerance that they found was 4 milligrams of oxygen per liter, because fragments that were exposed to less than 4 milligrams of oxygen per liter appeared to bleach and die within 3 days. And so considering that 4 milligrams of oxygen per liter does not even fall in that hypoxic zone that we previously discussed, which is less than or equal to 2 to 3 milligrams of oxygen per liter. This really represents the importance of further understanding the importance of hypoxia and the role it could have in impacting coral reefs, especially tropical coral reefs that we see with Acropora youngii here. And then this next study I wanted to highlight pertains to a cold water coral species, so more similar to the temperate coral species that I will discuss in our experiment, but this Hebeln et al. 2020 study looked at Lophelia pertusa, which I mentioned is a cold water coral species. And the oxygen tolerance that they found for this species was at 0.79 to 1.98 milligrams of oxygen per liter. So they actually found that this species was able to withstand very hypoxic water. And however, this was an observational study. And so while these colonies were found to be what appeared to be thriving in these low oxygen conditions, there is concerns over the actual experimental setup, considering that there was no experimental process in analyzing the presence of these corals. So it's unknown how long these Lophelia pertusa corals were present in this oxygen tolerance below, in this hypoxic condition, oxygen tolerance, as well as there's no assessment of the physiological responses of these corals as a result of the capabilities of being able to do an experiment like we did here. And so now to dive into our experiment, the model species that we looked at here within my honors thesis was Oculina arbuscula. And there are several unique things about Oculina arbuscula that make it a really great candidate for this experiment. The first is that while it is not a tropical coral reef building species like many people are familiar with, it's a temperate coral, so it can actually be found right here off the coast of North Carolina, as well as only found along the southeastern United States. But with that, it is not a reef building coral like many people are familiar with tropical coral reef corals. This coral, however, does still provide significant habitat for many local marine organisms. So it is an important habitat provider, similarly to those tropical coral reef building corals. And then the other unique thing about Oculina arbuscula that I'd like to highlight is that it is a facultative symbiont coral. And what that means is that Oculina arbuscula can live with or without a symbiotic dinoflagellate known as Symbiodinaceae, whereas most of the tropical corals that many people are familiar with rely upon that symbiotic dinoflagellate Symbiodinaceae, making them obligate symbiont corals. And so here on the left, we have our example of our coral fragment without that symbiont, known as an aposymbiotic colony. And you'll note that it's aposymbiotic because of the white color that you see. And so what you're actually seeing is the coral skeleton through the translucent tissue on this colony. And then here with our symbiotic colony on the right, we have the dark pigment, which is how you can tell that it's a symbiotic coral, because you're seeing the pigment of the symbiont or Symbiodinaceae through the coral tissue. And so now to dive into my hypotheses, the first, I asked one overall question really throughout this experiment, and that was, will hypoxia have an impact on coral protein concentrations? And most specifically, host protein concentrations, as that's what we looked into in this study. And my first hypothesis was that corals in the hypoxic treatment will have lower protein than corals in the normoxic treatment. And my reasoning behind this statement is I believe that corals that experience increased levels of stress, likely as a result of hypoxia, will experience greater protein degradation and produce less protein as well due to that increased level of stress. As well as with my second hypothesis, I proposed that aposymbiotic corals or colonies exposed to hypoxia will have higher protein than symbiotic corals. And my reasoning behind this statement is that I believe that these aposymbiotic corals, due to the lack thereof the symbiont or Symbiodinaceae, will experience less protein degradation and less reduction in their protein production due to a little less of stress as a result of not having to deal with the symbiont while experiencing this stress. As has been seen with other stressors like temperature, some corals, usually as a result of increased temperatures, will expel their symbiont and have costly effects as a result of their containing the symbiont. And so therefore I thought that the lack thereof the symbiont would produce less stress on the coral, and then those colonies would therefore have higher protein. And so how did we look into this? We had an experiment that had two treatments, the first being a normoxia treatment at 6.40 milligrams of oxygen per liter. And then we had our hypoxia treatment, which was at 2.7 milligrams of oxygen per liter. So you can see it's falling within that 2 to 3 milligrams of oxygen per liter range. And then in total, we had 9 aposymbiotic genotypes and 9 symbiotic genotypes. One fragment of each genotype was then exposed to our normoxia treatment, and then one fragment to our hypoxia treatment. And so in total, we had 36 fragments in all throughout the course of our experiment. And these fragments were exposed to these experimental conditions for a total of 14 days. And then finally, to carry out this experiment, we use the Neptune Apex fusion system, which is a system that allows us to monitor and maintain our environmental parameters. So in this figure or picture on the left, you'll see these modules up at the top of this rack here. Those are the modules from the Neptune Apex fusion system, and each module corresponded to one tank. So we had temperature and dissolved oxygen probes in each tank, and they would take measurements of our environmental parameters every 10 minutes. So we had that throughout the course of our experiment, as well as the measurements from the dissolved oxygen probes in our hypoxic tanks were used to maintain our dissolved oxygen levels around hypoxic conditions. So we were able to program the system to administer nitrogen gas, which was what we used to displace the oxygen in our hypoxia treatments. And it would therefore administer that nitrogen gas when levels got above about 1.6 milligrams of oxygen per liter, and then would stop administering that gas when they fell below 1.4 milligrams of oxygen per liter. And so now going to the post-assessment, or what we used as a health metric throughout this study. Like I mentioned with my hypotheses, we looked at host protein. And what was unique about our experiment was, since I was doing this project in collaboration with Bryce Corbett, a master's student in the lab of Dr. Fogarty, to make sure that we did not compromise the samples for her experiment and for my experiment, we had to go through some trial and errors in developing a new extraction procedure, as opposed to the procedures previously used in Dr. Fogarty's lab. And so I'll kind of describe it here quickly for you. What we did to extract the protein from our samples is, at the conclusion of the experiment, samples were airbrushed strictly with air to get tissue off of the coral skeleton. And then once we had the majority of the tissue off of the coral skeleton, we took that tissue and put it in this mortar and pestle that you'll see here up in the upper right-hand corner, and put liquid nitrogen in there with the tissue so that we could grind that tissue down into a frozen powder. And once we had the tissue in a frozen powder, we then put that in an Eppendorf tube where it was suspended in RIPA buffer, which is a buffer used to prevent degradation and preserve proteins. And then once in our RIPA buffer, we further homogenized our sample using sonication, which is this bottom picture that you see here. And then prior to actually taking and performing our Bradford assay, we centrifuged the samples to remove the symbiont or Symbiodinaceae into a pellet, as well as our cellular debris, and therefore took the supernatant of that sample to perform this Bradford assay. And the Bradford assay, we used a SpectraMax M2 to read the absorbance values with a Bradford dye. And those absorbance values were then compared to a standard curve that we ran to determine the protein concentrations throughout our experiment. And so, since this was a new procedure that we had developed, we had to ensure that we were looking at just host protein. And so to do so, I used microscopy to look at both the pellet and the supernatant of our samples. And these two pictures here that you're seeing are pictures I took from the pellet of my samples. So this first one here, you can see several Symbiodinaceae, which are kind of those little brown circles. Those are the symbiont or Symbiodinaceae cells. And here you can see them intertwined with some cellular debris. And then on the left, I have some symbionts or Symbiodinaceae that are alone, so you can get a better look at them. But what's important is we found that all of the symbionts that we found, or at least saw within our pellet samples, were intact, and we found no presence of either not intact or intact symbiodinaceae within our supernatant. Therefore, we are under the safe assumption that we are looking at just host protein throughout our experiment. And so now to dive into my environmental parameters. First, I will present temperature. So this figure shows the temperature throughout the course of the experiment in degrees Celsius. And on the x-axis, then we have our days of the experiment. And the two treatments that I have were hypoxia and normoxia. So you can see here in blue is my normoxia treatment, and then in purple is the hypoxia treatment. And so the goal was to keep the temperature constant throughout our experiment, as it was not a factor we were looking into. But you will notice that with our Wilcox unsigned rank test, we did find a significant difference between our hypoxia and normoxia treatment. However, because of the fact that the species we were looking into, Oculina arbuscula, is a temperate coral species, and the difference between our mean temperature for our hypoxia treatment and mean temperature for our normoxia treatment is less than 0.5 degree Celsius. We are likely not concerned about this difference because within the natural environment, these coral colonies can withstand temperature changes throughout the course of a year of up to over 20 degrees Celsius. So likely 0.5 degree Celsius is not having any major impacts on the coral physiology. But the likely reason we saw this difference is because the hypoxia treatment saw the addition of nitrogen gas to displace that oxygen. And since nitrogen gas does come in a pressurized form when we were administering it, likely since pressurized substances usually have higher temperatures, the addition of that nitrogen gas is likely why we see the slight increase within that treatment. And so now to look at our dissolved oxygen throughout the course of our experiment, we have dissolved oxygen here in milligrams per liter on the y-axis, and then our days of the experiment here on the x-axis. And what's important to note once again is that our Wilcox unsigned rank test did find a significant difference between our two treatments, which is good because we were able to ensure that there was a difference between the oxygen levels in our normoxia and hypoxia treatment. And with that, however, you will see there is quite some fluctuation within our hypoxia treatment throughout the course of our experiment. And that is likely due to some logistical constraints that we had when acquiring enough nitrogen gas to displace oxygen throughout the entire course of our experiment. So you do see some of these fluctuations where we ran out of nitrogen gas. But that likely is not too concerning as well, because throughout the course of a hypoxic event, we are likely to see some fluctuation in dissolved oxygen concentrations as well, whether it be through DL cycles or other things like tidal cycles. But overall, you will likely see some natural fluctuation. So this may actually be more representative of a natural hypoxic event with the fluctuations in oxygen levels. And so now to dive into my results on protein concentrations. First, we'll compare my hypoxia and normoxia treatments all together. So on the x-axis, we have our hypoxia treatment in purple and our normoxia treatment in blue. And then protein concentrations in milligrams per centimeter squared is here on the y-axis. And so this figure is going to test my first hypothesis, which is that corals in the hypoxic treatment will have lower protein than corals in our normoxic treatment. And with our Wilcox unsigned rank test, we found no significant difference between these two treatments, therefore providing no evidence for my first hypothesis that corals in the hypoxic treatment will have lower protein than corals in the normoxic treatment. And now to dive over to the second hypothesis that I proposed. We looked at protein across all 4 treatments. So here on the x-axis, I have my hypoxia exposed aposymbiotic corals, and then the normoxia exposed aposymbiotic corals. And then once again, the hypoxia exposed symbiotic corals, and then the normoxia exposed symbiotic corals. And along the y-axis, once again, we have protein concentrations in milligrams per centimeter squared. And so looking at the second hypothesis, which was that aposymbiotic corals exposed to hypoxia will have higher protein than symbiotic corals, we found no significant difference between all 4 treatments with our Kruskal-Wallis test, once again providing no support for our second hypothesis. So while we didn't find support for our 2 hypotheses, we did find something interesting that we initially didn't intend to look into, and that was with the protein concentrations between our 2 colony types. So on the x-axis, we have our aposymbiotic colony type, and then our symbiotic colony type. And here along the y-axis, we have our protein concentration, once again in milligrams per centimeter squared. And this time, our students' t-test indicated a significant difference between these 2 colony types, with the aposymbiotic corals having a greater level of protein concentration than the symbiotic corals. And so to get to our conclusions, first, we're going to look at the hypoxia versus normoxia treatments. And the reason we likely saw no difference between our hypoxia and normoxia treatments is the fact that Oculina arbuscula is likely a robust species, meaning that because it is a temperate coral species, it deals with large fluctuations in temperature, which can range from about 5 degrees Celsius to 28 degrees Celsius throughout the course of a year in this species range, as well as it's likely to deal with fluctuations in salinity. And so because of those other conditions which it deals with large fluctuations, dissolved oxygen may just be another factor in which it is able to withstand fluctuations in its natural environment. However, another reason why we may have not seen this difference between our hypoxia treatment and normoxia treatment is the role of a diffusive boundary layer. And so a diffusive boundary layer is a layer right around a coral skeleton where we see decreased levels of certain solutes. And oxygen being one of those solutes would have a decreased level of its presence or concentration right around the coral skeleton as opposed to the surrounding environment due to the diffusion of oxygen across the coral tissue for respiration. And one of the large factors that has a role in the occurrence of a diffusive boundary layer is water flow. And so here on the left, you'll see what a diffusive boundary layer is likely to look like with lower levels of water flow, as opposed to what a diffusive boundary layer would look like with higher levels of water flow. And so within our experiment, we did have power heads that we used to ensure that we had homogeneous oxygen levels throughout our aquaria. And this may have actually compromised our experiment as a result of that increased water flow, which with those power heads was anything up to 98 centimeters per second. And as a result, we may have seen the diminishment or a lower level of a diffusive boundary layer, which could decrease the effects potentially of hypoxia, as if hypoxia is present in the natural environment and you have the compounding factors of a diffusive boundary layer, seeing that corals are prone to develop them as a sessile marine organism, that could actually enhance the hypoxic effects that corals would experience. And so because we saw such large levels of water flow with our power heads, this may have diminished the occurrence of this diffusive boundary layer. One study actually found in boulder corals that as much as a 5 centimeter per second presence of water flow was able to diminish the presence of a diffusive boundary layer by as much as 25%. So if you think to increase that about 20-fold, we would likely see vast decreases in that diffusive boundary layer. And now to look at the aposymbiotic versus symbiotic colonies, which, as a reminder, we saw an increase in protein concentrations within the aposymbiotic colonies as opposed to the symbiotic colonies. The reason we likely saw this is due to the reliance on heterotrophy. So these 2 coral colonies are likely to have slightly different lifestyles that contribute to this. And the aposymbiotic colony, due to its lack thereof, the symbiont, must rely solely on heterotrophy to meet its metabolic needs, whereas the symbiotic colony can also depend and likely supplements majority of its coral nutrition through the byproducts of photosynthesis from its symbiont or Symbiodinaceae. And so as a result, not only, and I'll play this little video of Oculina fragment feeding in our lab here, but not only are these coral colonies likely to have higher levels of um, protein or nutrient allocation proteins, but also digestive proteins. But as well, another major factor may actually be the supplements or metabolites that these coral colonies are uptaking. And so the Oculina arbuscula colonies are much more likely to uptake protein-rich metabolites because they are feeding on different things within the water column, whereas our coral colonies that are symbiotic are likely to meet most of their metabolic needs through photosynthesis, which would provide sugar and starch as a byproduct to meet most of their metabolic needs. And so if you kind of look at this figure again, you'll see these tentacles of this polyp are kind of very active in as an aposymbiotic colony in pulling out the reefroids that we were feeding this colony, as well as some real oceanic eggs. And so you can see kind of the occurrence and how that would actually have a role in the enrichment of the protein that this colony would be taking in. And so now to get into the significance of this experiment, the first is the importance of Oculina arbuscula, and most specifically coral in general, as a habitat provider. And so the lack of these colonies or any negative consequences that corals would face would have devastating impacts on not only for tropical coral reefs, but also here for local marine systems as a result of the loss of this habitat that they provide. And so these cascading ecosystem effects are important to understand, and it's important to understand what effects climate change has on these important habitat providers. And then the other significance here is the interesting thing that we were able to do by looking at a facultative symbiont, which allowed us to look into that host symbiont relationship and if there were any important implications there in looking at how hypoxia affected the occurrence of that host symbiont relationship. And this could have impacts and be important to understanding how obligate symbionts, what threats they face since they require that symbiont and don't have the option to live without that symbiont. And so there are many important implications to take away from that. As in the case of this study, it may show that there are no significant consequences to living with or without the symbiont in response to hypoxia. But more research would also help in further investigating that theory. And so finally, I have many people to thank throughout the course of this year for help with this experiment and through my honors thesis. First, I want to extend a large thank you to Dr. Nicole Fogarty for serving as an advisor and being a part of my committee and providing valuable advice throughout this year, as well as once again, my honors committee in Dr. Chen and Dr. Tift for providing advice and feedback throughout this year, as well as being here today to help me execute my defense. And as well, I'd like to thank Bryce Corbett, the master's student that I worked alongside throughout this past year, for serving as a mentor, but also allowing me to use a piece of her master's thesis to look into this experiment. The CSURF office here at UNCW for awarding me a research supplies grant to help fund some of this research. I'd like to thank Dr. Kinsey's lab for help with developing that new extraction technique that Bryce and I were able to develop, as well as providing us with some of the equipment and procedures that we were able to use, as well as the Reef Lab for anything from the experimental setup to experimental analysis. And finally, all of you for joining and coming here to listen to my research and what I've been doing for the last year. So thank you all, and I look forward to your questions.
Thank you very much, Ryan. All right, at this time, we're going to open it up to anyone who has questions for Ryan. So you can type in the chat, you can raise your hand, you could just unmute yourself. So questions for Ryan?
I can start it off. That's okay.
Yeah, go for it.
So Ryan, thanks for that great presentation. And I'll save questions for later when we meet, but do you find the symbiont and the asymbiont in the same temperature in the wild?
So in terms of the temperature, I'm not sure specifically on the differences there, but what is likely is that usually the aposymbiotic corals are found in more shaded areas or deeper areas. So it likely is a result, while it's not specifically likely due to temperature, but more the association of light. But that could also have a role in the temperature that those corals are experiencing.
And then do you have evidence of metabolic rates with temperature between asymbionts and the symbiont as far as oxygen consumption rates?
I do not have any current data supporting or looking into that, but I think that would be something really interesting to kind of further look at these metabolic rates and the associations there.
Okay, thanks.
Thank you.
Other questions for Ryan? Don't be shy. Going once. Twice. Anyone? Okay, if no one.
I have a question, sorry.
Yeah, no, please. Go ahead, Mia.
Ryan, do you think that the Oculina may play a big role in coral restoration in the future?
In terms of coral restoration, I don't think they're going to have as much of a role in the traditional aspect that we think of coral restoration. I think the findings from this study could be important for understanding coral restoration, but in terms of tropical coral reefs, which many people associate with coral restoration, I don't think they'll have as much of a role, as they're not a tropical coral, so they're a temperate coral, so you wouldn't really use them for transplanting on tropical coral reefs. Thank you.
Any other questions?
Ryan, I have a question. I just was wondering, if you were to do this experiment again, what is kind of one of the biggest things that you would do differently?
Definitely. So I think personally, the big thing, kind of as I talked about that diffusive boundary layer theory, would be finding a way to make sure that we had homogeneous oxygen levels throughout our aquaria, but really kind of keep that in check and see, look into the difference of maybe water flow in this study as well, and how that would have a role in that theory potentially impacting the corals in terms of if hypoxia was able to be enhanced through diffusive boundary layers.
And is there a way to measure the diffusive boundary layer around a coral? Do you know?
There are ways. I know there are microsensors that you can use to get and kind of closely monitor levels as you get closer to the coral skeleton. So you can kind of use these microsensors to then measure oxygen levels across a profile as you get closer to that skeleton. So you'll be able to kind of then create a model of what that diffusive boundary layer looks like. And there's some other techniques as well, but I believe that's the most common one that people will use.
Interesting, thanks.
Anyone else?
I have one question.
Yes, Blake.
That was a great presentation, by the way.
Thank you.
I guess in terms of there's this whole thing about breeding supercorals and engineering more robust corals. How do you see your research fitting into that, and what are your thoughts and how this can go?
Yeah, I think in terms of this research, I think the important part is kind of first, and looking in that host symbiont relationship and really seeing what role that has. Will facultative symbiont corals, is their presence and ability to kind of live with or without the symbiont, is that beneficial to them? Or in responding to hypoxia, or is that not a great concern in looking at coral reef restoration and kind of developing, I guess, a better coral line? But also just in terms of coral reefs, because I think, like I said earlier, a lot of people think of tropical coral reefs when they think coral restoration. This could have kind of greater understanding in just hypoxia overall, and most specifically on that host symbiont relationship and how that would pertain to them, since most tropical corals are going to require that symbiont. Thank you.
Any final questions for Ryan? All right, if there's no other questions, let's go ahead and congratulate Ryan for a nice presentation one more time. Well done. And at this point, the open portion is completed. And so we'll have you guys sign off. Drs. Chens and Dr. Tiff, please stay on for his closed session. Thank you for joining us.
Thank you all. Thanks for coming.